pleasure. It's an honor to have here Nelly Sélem Mojica. She is a researcher from the Centro de Ciencias Matemáticas UNAM, so from Mexico. Nelly is a Mexican bioinformatician and mathematician interested in big data, in metagenomics, and genome evolution in prokaryotes. After a PhD and a postdoc in integrative biology at Langevio, she's starting her research group at the Center of Mathematical Science in UNAM Morelia. She has developed genome mining tools for which she earned the Mexican L'Oreal Award for Women in Science 2021. Congratulations. And she's a member of the Mexican SARS-CoV-2 Genomic Surveillance Consortium. So she likes to contribute with software education resources, such as a lesson for the Carpentries Incubator and Wikipedia content. In other interests, and as an entrepreneur, she's a founder, member of Better Lab, a biotechnology and software startup, and personally, and she cares about statistics for women in sports. Teamwork, family, and social contribution matter. She looks for a life-work balance for her and her team. So as you can see, she's a really inspiring model for us. Thanks a lot for being here today, and welcome, Nelly, and the stage is yours.
Hello, everybody. Thank you for the presentation. I'm glad to be here, and I'm going to share with you what I am doing right now. I'm going to talk to you about MicroAgrobiome, a comparative metagenomics platform to study Clavibacter in the microbiome of Mexican crops. First, as you have said, I am a mathematician, and also I ended in a PhD biology. So mainly I have worked with genes and their function and how can we gather these gene families. So I have developed tools for find gene families involved in antibiotic biosynthesis, and also to find marker gene families to distinguish between close species. For example, if we have a species A and these are gene families, we can see that this family that only belongs to species A allow us to differentiate from species B. This other family marker 2, is only present on species B, so we can differentiate between a genome from species A and a genome from species B. So this is where I was working while I was on my PhD. And now that I am starting my group, so I don't have too much results to present today, but this is what we have been doing this year. I have continued with these ideas, but on metagenomics. Why on metagenomics? Well, because microbiome public data is quickly growing. When I started my PhD, my lab mates maybe have 10 sequences for some gene family in a taxonomic lineage. When I ended the PhD, I ended with 3,000, 4,000 sequences of the same gene family. So I leave this public data grow, and I was very happy that I can work with such a big data during my PhD. And the same is happening now with microbiome public data. The term microbiome was used for the first time in 1998 by Joe Handelsman. And for example, in 2005, there were studies that only use one metagenomic data, 2, maybe 5, but then now in 2020, 2021, we can see studies that uses 1,000 of metagenomic data. And there are even also some studies that uses 10,000, 20,000. So we are really now on the microbiome big data time. So this is why I am excited to work with microbiomes, and also because public data allow us now to answer general questions about microbial ecology. For example, I think 3 key questions in microbial ecology are: where do microorganisms live? Which microorganisms and which are the the interaction of these microorganisms, and also what is the microorganisms' metabolic potential. So where, with whom they interact, and what do they do? Well, but there are a lot of environments, so we have to center in some of them to really get to the details. I am interested in soil microorganisms. Recently, a global atlas of soil microorganisms was published. And as we can see, there are more than 20,000 phylotypes that they have found, but only 2% of them are present in most of the soil samples that are studied. This 2% explains a lot of the abundance. They are not only present in most of the samples, but the abundance is really covered by these 2% phylotypes. And who are these phylotypes? Well, there is, of course, proteobacteria and actinobacteria among the most ubiquitous microorganisms in soil. Okay, but soil and rhizosphere are also different ecological niche. Why? Well, rhizosphere is the part of the soil that it is close to the root, and it's different from another soil sample for other region. So now we can go to the question, which organisms are present in crops rhizosphere, not just in any part of the soil, but in the crops rhizosphere. So I choose some of the relevant Mexican crops, some of the crops that we eat all the time, like corn, potato, pepper, tomato. And I have noticed that there is a genus, Clavibacter, that it is present in all of these crops. Clavibacter is an actinobacteria, but it is not present in all of the soil samples. It's not a ubiquitous genus. But this genus have species in all of these crops. For example, Clavibacter michiganensis subspecies tessellarius is a species that colonizes wheat. And Clavibacter michiganensis subspecies nebraskensis is a species that infects corn. But today, I want to go deep in the details of Clavibacter michiganensis subspecies michiganensis. This species is a tomato pathogen. And once that you get infected by Clavibacter michiganensis subspecies michiganensis, the plants get they die. They really quickly die on the greenhouses. There is a total loss, and it's really of economical importance, this microbial pathogen. So when you find Clavibacter michiganensis CMM on a greenhouse, all that section is burned, and you want to get rid of Clavibacter michiganensis. So it's like in SARS-CoV-2, everything we were burned. Then where does this pathogen live? Because I don't know where CMM lives when it is not causing a tomato infection, you know? Does it live on the water? Does it live on the soil? Does it live on the seeds on another plant? Does it live on the rhizosphere of another plant? Does it live on the grass that it is in contact with tomato? Where does CMM live? Well, we don't know, but what we can do now is to go and look on the public data, and we have observed that the Clavibacter genus is present in low proportion at cultivated plants rhizosphere. For example, these are several pepper rhizosphere samples and several potato rhizosphere samples. It is a rare OTU. It's a rare species present, but it is there. It is always there at 2%, 3% of the reads of the total reads of the sample. When you go to the traditional bioinformatic pipelines, you get that Californiensis, Capsicisi, Chilensis, you get several species of the Clavibacter genus that the reads are taxonomically assigned to this species. These are world public data from 2021. Nevertheless, species level assignation is not highly reliable for rare OTUs. Why? Because if you only have some reads, these are the reads, the dashed lines, and these are a genome a genome B. Well, if you only have some reads and the two genomes that you are trying to distinguish are really close phylogenetically, well, maybe the reads that you have are on the shared regions of the genome. And if you do not have enough SNPs or enough variation on those regions, then you are not going to be able to distinguish between these two species. Maybe marker gene families can help with these species level assignations. If by any happy coincidence happens that you have a read that maps to a family marker region, a region that it's only for the species A but not for the species B, then you are going to be more confident that the reads that you have are for this species. You are going to be more confident that you are finding Clavibacter michiganensis and not Clavibacter michiganensis nebraskensis, for example. But how can you get to know to these families? Well, pangenomes can help us to localize marker gene families. We can gather all the genomic information available even in public or private data. And we can look on the intersection of all the Clavibacter michiganensis genomes. And this is a family that is going to be present in all the michiganensis genomes. And also, you can make sure that it's not present in the union of all the other Clavibacter species, not in nebraskensis, not in tessellarius, not in capsicisi. And this is the software that I have showed you at the beginning. So you can localize gene marker families using pangenome information. And hopefully, some of the reads are going to allow you to discriminate between michiganensis and nebraskensis if they map to these families. So we constructed a pangenome, a Clavibacter genus level pangenome with 84 genomes. Here I am showing you all of the genomes. These orange ones are Clavibacter michiganensis. These are grass-associated species, and these are the other species of the genus. As you can see, we found some marker gene families, but nevertheless, until now, the reads that we have been looking for do not map to these families. So we have to think of another strategies to really discriminate between genera, between species, and in that way, we can at the end answer the question of where CMM live when it is not infecting tomato. Well, in the other side, with this pangenome of Clavibacter, we constructed a multi-locus phylogenetic tree, and we have noticed that the grass-related Clavibacter are really close to tomato hosted CMM. So it is possible that maybe Clavibacter michiganensis lives on the grass and then goes a jump into tomato. But this is an hypothesis that it's still have to be tested. This is the work of Alan, and I hope it's going to be published soon. Then we added 37 Latin America rhizosphere metagenomic data, just to see if in this data we can find some of the marker gene reads. We work with chili, corn, tomato from 2021 and 2022 from Sinaloa, Aguascalientes, Peru, and Guanajuato, but no. We still finding consistently reads that map to Clavibacter. The software says that it is Clavibacter michiganensis, but we are not sure at the species level. Some of the reads align to plasmids and some to the main chromosome. Well, we are going to keep looking where does Clavibacter live. But then we question ourselves about the interaction networks. We center the cluster in Clavibacter, and we look for the interaction network in the samples of chili, corn, and tomato. As we can see, in red is actinobacteria, and the other colors are some kind of proteobacteria, the most abundant phyla in rhizosphere. And the tomato network is the smallest one, and there are less interaction in tomato, seems, than in the other crops. And what we found is that two genera, Bifidobacterium and Curtobacterium, are always positively correlated with Clavibacter. So when Clavibacter grows, also grows Curtobacterium and Bifidobacterium. This happens in all the three crops, corn, tomato, and chili. And here you can visualize this result. Always when there is Clavibacter, is the orange one, there is an increasing of Curtobacterium. Clavibacter, Curtobacterium. It doesn't matter if it is in tomato, corn, or chili, or the geographic location, this happens. It could happen because Curtobacterium eats the dead tissue of the plant, but this is a result that we found only bioinformatically, and we have been able to corroborate this in the literature. Well, to take home messages from this part is that Clavibacter genus is more distributed than previously thought. Reads that map to Clavibacter were found in Mexican rhizosphere metagenomic data. And also there is Clavibacter genus reads in Cuatro Ciénegas, in water samples, even in the inside of some mouse, some reads that map to Clavibacter have been found. So Clavibacter is more ubiquitous genus that we have thought before, but it is not completely ubiquitous in all the soil. The reads that we found, some map to plasmids. No CMM gene marker has been found yet in those reads. And also Curtobacterium and Bifidobacterium correlates positively with Clavibacter in corn, chili, and tomato. This is what I have to tell you by now about Clavibacter, but now I want to summarize all these results talking about bioinformatic platforms. So we really loved our work to be repeatable and shared with other people. For that we have developed this lesson for metagenomics to learn metagenomics, and we have a learning server free to use. If you want to use it, you can ask for that. In fact, the next workshop, we are going to be using this lesson and our server. But this lesson is just to start learning the alpha index of diversity, the beta index of diversity, and the taxonomic classification. But after that, there is a lot that we can do, as we have been talking now. That is why we have been developing MicroAgrobiome. And in this platform, we want to gather information of the rhizosphere microbiome of relevant crops, at least for Mexico. And now we have public data of maize, tomato, chili, wet potato, and alfalfa. And what we want to do is with the abundance tables, we want to compare new samples with stored data. We want to identify similar metagenomes. We want to then gather these genomes, cluster them by temperature, geographic localization, and other metadata. And after that, we want to construct the co-occurrence network of especially centered on some microorganism of interest, and to give a profile of health of the rhizosphere. And by that, I mean to the identification of pathogenic and beneficial bacteria. Finally, we want to integrate some metadata analysis like the health crop, and when possible, add a pangenome for selected microorganisms to obtain detailed information. So until now, I have been talking to you of this mixed kind of data, amplicon, 16S, metabarcoding, and also shotgun sequencing. So I mean all the genome sequenced, and also only the 16S sequenced just to know who is there on that sample. But these are, we have these two types of information on the database. So I want to finish this talk with the MicroAgrobiome Santa's letter, what really I would like it to do. And it is to get metabolic information from the shotgun reads. At the beginning, I let you know that my other interest was to find genes that work on finding antibiotic information. And for that, I am using the information of main copy and secondary copy. So I need genome information. Here, what you see is a gene family. This is the primary copy of that gene family for each genome, and these are secondary copies. This software finds novelty in the pangenome data, but using copy number per genome information. When you have metagenome data, you only have some reads. You don't have all the genome information. So in metagenomic data, we do not have copy number or other genomic information. Nevertheless, the trees, okay, they are visually insightful, but there are other representation of distance between sequences. At the end, the trees, the data are dots, and other distances may be adapted to this Evo mining to metagenomic data. How can we do that? Well, for example, we can apply machine learning algorithms such as support vector machine to divide between conserved copies and evolved copies without the need of the genome copy number. We can also cluster by some other distance to divide conserved and specialized genes. So current evolutionary genome mining starts from a genomic database now, but we are losing all this metagenomic information. So maybe for the second part of MicroAgrobiome, I would like to also add a pipeline like this. You start with a metagenomic reads, then you binning these reads into a lineage, then you apply classification algorithms, and you ended up with a visualization of conserved members of a gene family and the ones that could be now devoted to antibiotic biosynthesis. So if comparative genomics has taken us so far, why not think about comparative metagenomics? Now is the time of comparative metagenomics and comparative pangenomics. There is much more information there, and it is available for us now. So I started telling you that I have worked on these two types of software. So now it's not me, it's we. We are developing tools that attempt to find gene families involved in antibiotic biosynthesis, both in metagenomic data, and to find at the species level the behavior of selected crop rhizosphere microorganisms. So thank you very much, especially to you for being here now, hearing my talk. And my genomic thanks to my PhD laboratory, Evolutionary of Metabolic Diversity, to Alan Yanez, Amber Gomez, to Dr. Francisco Barona, with whom I work the genomic part of the Clavibacter story. Alan is the one that is conducted that research now. And also I want to thank to my team and my all my collaborators now, especially to Fernando Fontove, Diego Garfias, Claudia Sirion, Abraham Avelar, Shaddai Guerrero, Solena, who provided us with the Latin American data, to the UNAM, of course, I'm very happy to be there, Better Lab and C3, and to CONACYT for the grants that they provide. Thank you very much, and I am open to questions.
Thanks a lot, Nelly. Amazing. A lot of things, not only research, but leading, developing tools. So such a variety. We have time, and we have, I will read you a question from the chat. Leda said that in the data that you use, they consider the milpa crop because chilled corn, green tomato, tomatillo, and chilacayote or pumpkin.
Well, the milpa, you have to cultivate all that plants together, and I have not access until now to a milpa, a traditional crop by now. We are planning to do so. We are trying to get those data, but no, until now we only have microbiome data of tomato, and then data of corn, and then data of chili separately. But yes, we would love to have milpa microbiome data.
Thanks. Juan Diego said, congrats, what an amazing work. No question. I have some questions, but some of them you answered to me during the presentation, but very basic one. When you define a OTU, how do you define it, or species or the genre? Do you define what similarity of the sequencing? How do you define that they are similar enough or not to be considered a new species? Something very basic, sorry.
No. We are using Kraken default parameters. Kraken is this software assignation tool, but I don't think that anybody really believes when Kraken say this is for this species. Well, maybe if the reads are abundant from some OTU, well, you have more confidence because Kraken compares by k-mers and they have to be exact match. And if you have enough, well, maybe it's okay and you can be confident that is that species. But if you only have a few, I think you can only hope for a genus assignation.
But this is using the metagenome reads, not the 16S, no?
Yes, metagenomics.
And then from the 16S, you have to do the same. It's like by similarity, you define that you have a new species in this metagenomic study.
By similarity, yes. We are not using yet the single variants. We are just using a threshold for similarity.
Okay, there is some extra question. Lila Rodriguez Perez said, what kind of tools did you use to align the reads to your gene markers, BOWTI, BWI, or other more specialized tools?
Yes, BOWTI and some tools, just traditional tools that are available. We have not developed anything particular for this part.
You develop a lot. You're doing a great job. So any other question, I think, in the chat? A question from YouTube. No, the same one. Which tool do you use for alignment? Okay, there is. I think there are no more questions. Interesting. Thanks, Nelly. Yeah.
In that sense, we are trying to construct a SNP variation library. So maybe it's not going to be about mapping the reads in some genes, but we are going to have to down one level and look into the single nucleotide variation. And also we are thinking about topological analysis of the data. This is the two directions that we are trying now.
Okay, thanks a lot. I think we are on time of closing the session. It was a pleasure to have you here.